Different populations.
Yeah.
Do you think that I should do exposed control stuff? Because I kind of have that for Carmel already. I mean, like, how many pie charts do I want to, like...
Okay, now we're done with pies. We're done with pies. Okay? We're,
Yeah, but you could do pie charts.
you could do it for each one. No. Now we're going to do something. Now we're going to do the comparison part. Okay? So what do you want to compare?
San next to Carmel's.
Infected
Disease?
them?
No. Well,
Four. Yeah.
I guess both of them infected, because what I'm looking for is disease resistance genes, right?
Yeah, or you could compare all four libraries. You have 4 libraries. You could just compare 4 libraries. So basically, you go down to CLC. All this is done in CLC, and you'll do it several times. But essentially, this file right here, that's the consensus of San Nicolas and Carmel, 584. So this is a FASTA file of all your contigs. Okay? From your assembly that we use to blast. We're going to use that as the backbone to do RNA-seq analysis. Okay? And RNA-seq analysis is just a fancy word for talking about how, looking at the different differential gene expression between them, okay? Okay. So these are your files that have been quality trimmed, all right? And then what we're going to do is we're going to do an RNA-seq analysis with the, nope, do one at a time.
So you do that one to the de novo one, and then the second one to the de novo, and you do them all individually?
Yep, and this is more just a way that the software works. So what it's really going to...
Does anybody besides you know how to do all this stuff in here?
Everybody in my lab better know how to do this.
Okay. Sam does?
He should, yeah.
What all your minimum, maximum shit is?
There's no magical stuff here. Basically, we really just do it, and if we don't like it, we do it again, and we talk, you know.
Okay.
So the maximum number of mismatch. So I'm gonna set it to defaults. The maximum number of mismatches is going to allow 2 mismatches. Length fraction, maximum number reads per hit, 10.
Didn't you change that?
This is what it is now. So we're just going to leave it at that. I've done it tons of different ways. It doesn't much matter. Can't even do anything there. Can't even do anything there. For organization purposes, I just put it in here. Finish. We just do the same thing. So what it's going to do is you know, take all your little baby reads, they're cleaned up, and map them back onto this common backbone.
Yep.
With.
Can you do all 4 at once?
Yeah, you can. No, you can, but it's not the point. So now you can have 4 separate expression files, and you can compare them. You could do them all together, but then you just have one expression file. Okay, so these will take a while. Well, I don't know how long they'll take actually, but a while. These are running on the big computer in the lab.
Okay.
And then it's actually quite easy at that point. And then what happens is you just, what they do is called create an experiment, where it just puts those together and does comparison and statistics and see which genes are differentially expressed. And we'll see if I can find one of Mac's things. So you're gonna have these two. Really, these are what you're gonna end up with. Are these RNA-seq files? So let's just pretend.
I'll have 4 of them.
You'll have 4 of them, but just to keep it simple, then we just do something like setting up an experiment. Well.
So this is actually what you want to compare.
Yep.
Okay.
And we can do all 4, and then what you can do is you're gonna get a file that like this.
Mm-hmm.
That'll have your feature IDs, which will yours be called like contig 1,
Mm-hmm.
and then a bunch of other crap, how much fold difference there are, and then you'll get some statistical numbers and.
She actually has his session numbers because they match.
She used a EST database. Okay. But see, you're going to have these contig numbers, right?
Mm-hmm.
And now you're going to have these other files that we generated a few minutes ago.
Where I know what the contigs are.
So then that's when all the magic happens. All right?
Okay.
So we'll stop there for today.
Okay. Yeah.
Does that make sense? Does that make sense, though?
I think tomorrow I'll do all this stuff tomorrow.